In this video, we're going to be looking at the ampular organ and the macule organ, just to go back and understand a little bit where that is. This is the entire inner ear structure. We've already talked about the cochlear structure. We're now going to talk about structures over here. Remember that there's an outer region of your inner ear structure that is filled with what we call a perilymph, and then within the inner structures, there's the endolymph. And there's a membrane that surrounds that endolymph here. It goes around this swollen spot at the base of each semicircular canal. There's one at the base of each, so there's three of them. These are called the ampular organs. So these ampular organs are at the base of our semicircular canals at one end. So there's one semicircular canal, and here's another one. There's the ampular organ, the swollen part, and then the tube, the duct that connects, goes all the way around. And then here's the third one. And it's important to note that each of these ducts of the semicircular canal is in a different plane. So there's one that's running this direction, kind of in the plane of the board. There's one that goes back, and then there's one that goes up. So we have three of them here. Each of these spaces is connected to the utricle. Another space that's inside of this whole structure here, remember that's called the vestibule. So here's the utricle structure down here. And that it connects then to the saccule structure over here. And then that connects to that cochlear duct that runs out through the cochlea over there. So what we are going to be looking at are structures found within the ampulla and in the utricle and saccule here. So right there, that's an ampulla. So we'll be looking at the ampular organ that's in each of those. And then within the utricle and saccule, there will be small structures, small little spots on the wall that help us with our static equilibrium, and that's referred to as a macule organ. So we're going to have an ampular organ inside the ampulla and a macule organ inside the utricle and saccule structures within the vestibular region of the inner ear structure. Now we're going to look at the detail within the ampular organ. So here, this was vestibule over here. Here's the beginning of our semicircular canal. At the base of this semicircular canal, we have this ampular organ. So the endolymph space that passes through here. Inside here, we have this modified structure. There's a large bump I'm putting in here. That is our organ for dynamic equilibrium that helps us know when our body is in motion, where we're going to experience some movement of our body and our head by that organ. Now within that, there are cells. Let me put those in a different color here. I'll just draw a few of them in here. These are the primary sensory cells called hair cells. And the hair cells have a long kinocilium, one long kinocilium on each hair cell, and then next to that they have some shorter stereocilia. They're involved in the sensory process. Now between the sensory cells there are usually some supporting cells. So I'll draw in a couple of green supporting cells in here that are associated with this layer of cells that form that organ of sensation for dynamic equilibrium. Near the base of those cells, let's see, we'll need to do another color. we encounter the nerve endings that help us to carry that information into the brain, our knowledge of our movement, our body movement, that dynamic equilibrium. So we have nerve fibers connected here to these cells. So what happens here is when your body moves, we have a movement of fluid within that structure. And whenever this flap-like structure sticking up here is moved one way or the other, it's going to cause a deflection of the kinocilium and the stereocilia at the end of each of the sensory cells, the hair cells in there. And when they are deflected in one direction, they reduce the flow of sodium into the cell, and that reduces the amount of neurotransmitter that's delivered to these nerve endings here. So it lowers the amount of signal to the brain. If it's deflected in the other direction, then it increases the flow of sodium into the cells. These hair cells depolarize and release more neurotransmitters to carry that signal into the cell. So the whole thing here is referred to as an ampular organ. A single structure can just be referred to as an ampulla. Within that, the flap-like structure we're looking at here that sticks up, it's kind of a gel-like material, not a cellular material here, and an extracellular material right there. And this whole thing sticking up is referred to as an cupula, And my marker's not doing too well. Cupula. It's kind of a generic name for things that stick up off the top of something. It's the same name used for steeple-like structures that stick up off of buildings, like on some horse stables. You often see those structures, and other kinds of buildings. At Churchill Downs, those spire-like structures that stick up there are referred to as cupulas. So anyway, this cupula, this little flap, moves back and forth. Inside there we have the hair cells. I've indicated in purple here. Hair cells, and they have a kinocilium. Kinocilium that sticks up, and then they have shorter stereocilia. In there. So we have, those are the hair cells. Let's see, and then we have supporting cells. Sometimes called sustentacular cells, and then we have our nerve fibers. These nerve fibers are connected to your vestibulocochlear nerve, the 8th cranial nerve, and they connect to a ganglion cell. They have a ganglion cell nearby that relays that information to the brain. So that's how our sense of dynamic equilibrium works. If we get movement in one direction, you increase the number of signals going to the brain. You move it the other direction, you decrease the number of signals to the brain. We have them in 3 dimensions, 3 different directions, 3 different planes that we can detect movement in. And we get one kind of signal from one side of the head and one kind of signal from the other side of the head. So we know which direction as well as which plane we are moving in for any movement of the head. So that's our sense of dynamic equilibrium using our ampular organ. I forgot to mention that in this ampular structure, the specific part that does the sensation responsible for the conversion of the physical signal to the neural signal, all of this structure here, let me put a dotted line around it here in blue. All that part right there is referred to as a crista, or crista organ, the crista of the ampular organ is just that section that has the flap-like part, the hair cells, and the beginning of the nerve fibers. So it's kind of a transduction of the physical signal to a neural signal, and that's where it occurs, in that crista organ. So there's another term that's important in understanding the overall ampular organ for dynamic equilibrium. Now we're going to look at a macule organ. These macule organs, these spots, are found on the wall of the utricle and saccule portion of the inner ear apparatus. So this is within the vestibule, the vestibular portion of the inner ear apparatus. And within this macule organ, it's just kind of a raised spot. Macule means spot, so this is a spot organ or macule organ. In the macule organ, there are, again, hair cells, and the hair cells have a kinocilium and then some stereocilia. So there's one hair cell, here's another hair cell. They're columnar cells, and then they have the kinocilium and stereocilia attached to them, their apical surface. So here we have some hair cells in the macule organ. There are supportive cells for these also, kind of found around them and between them. So anyway, above the hair cells, there's another gel layer. So we have the gel layer, and then on top of that gel layer, we have small crystalline structures that the body produces that form a layer of these dense crystals on the surface. These crystals are called autoconia, and they usually fuse together to make kind of a solid mass, a dense mass here on the surface called an otolith. That means ear stone or hearing stone. Otolith, made up of all these little crystals, or autoconia, on the surface of the gel layer. Beneath that, we have the nerve fibers that come in here that make association with the base of the cells, very much like what we had with our sense of dynamic equilibrium. This is our sense of static equilibrium, so here are nerve fibers. Again, this is part of your 8th cranial nerve, your vestibulocochlear nerve. These fibers reach out to a ganglion cell somewhere just outside of this structure and then relay that to the brain. So the way this works is the force of gravity has a greater effect on the more dense material, on those autoconia on the outside. So they would cause this gel layer that covers this here to sag in one direction or the other. And so this is very much like our dynamic equilibrium function, and that movement of our hair cell cilia, kinocilia and stereocilia in one direction, give one kind of signal, and then movement in the opposite direction give it the opposite kind of signal. So this whole structure here fused together is referred to as an otolith. The individual crystals are autoconia. There's a gel layer. There are sensory cells or hair cells. And again, we have kinocilium, and we have stereocilia associated with each of the hair cells, and then we have supporting cells, and we have nerve fibers. So this is our sense of static equilibrium. We know where we are with respect to gravity. What's up and what's down is basically what this organ tells you. And they are situated in different planes on different walls of the utricle and saccule. So one would be in one plane, one in another plane, and so no matter where you are, you know where you are with respect to gravity. Gravity will pull on the denser material that gel, and the gel layer moves with the sagging of the autoconia to one side, and that causes a deflection of the hair cells, this kinocilium and stereocilia that extends up into the gel layer. When they deflect in one direction, they depolarize and increase the signal going to the brain. When they go the opposite direction, they hyperpolarize and reduce the amount of neurotransmitter released, decreasing the signal to the brain. So that's your sense of static equilibrium found in the utricle and saccule using these, what we call macule organs. So this is the macule organ, the entire structure.